welcome to Expert Guidance, and today in this video we'll be covering the topic 4.2 Organization of your AQA GCSE Biology Paper 1. So just to remind you, for your Paper 1 chapters you need to cover the cell biology, organization, infection and bioenergetics, and this video is focusing on the topic of organization. Now in this video we'll be looking over what is the organization hierarchy of a living system, human digestive system, human circulatory system, heart and the blood vessels, the blood, the coronary heart diseases, non-communicable diseases, respiratory system, plant tissues, plant organ system, and transpiration. So let's begin. Now, in the previous unit, we studied about the cell, which is the basic structural and the functional unit of the body. And the examples are the nerve cells and the muscle cells. Now, these cells, when the group of cells combine to perform a specific function, that becomes the tissues. When groups of tissues combine together to perform a specific function, it becomes organs. And when groups of organs combine together to perform specific functions, that become organ system. An organism is nothing but it's the collection of different organ systems. Now, what are the different organ systems? Like circulatory system, digestive system, nervous system, excretory system, they make up the different organ system of animals. And if we talk about just the digestive system, the different organs in that will be stomach, kidney, liver, pancreas, heart. These are the different organs. And then the organs are further made up of smaller tissues like muscular tissue, epithelial tissue, glandular tissues, and the tissues are made up of collections of cells. So you should know these hierarchy and the examples of them. Now, let us talk about digestion. Now, what is digestion? Digestion is the process of breaking down of large insoluble food particles in the food into soluble smaller particles which can then be absorbed in the blood and used up. Now, what are the bigger components which are present in the food? These are carbohydrates, proteins, and fats. The carbohydrates get digested by the special enzymes called carbohydrases into simple sugars. Proteins get digested by proteases into amino acids, and fats are digested by lipases into fatty acids and glycerol. Now, this is what is the structure of the digestive system, and you should know how to label it. So it starts with the mouth and the pharynx, and then there are the salivary glands that releases saliva which softens and moistens the food, and saliva also contains the enzyme amylase that begins the process of digestion of starch to sugars. So you must have seen people says that if you chew the food, it tends to taste sweeter. The reason is that the amylase enzyme, it starts to digest the starch into the food into simple sugars. Then the food is pushed down into this food pipe called esophagus, and then it reaches the stomach. In the stomach, the hydrochloric acid is released. This hydrochloric acid first kills all the infections which comes up with the food, and it also makes the pH acidic so that stomach enzymes can work, because stomach enzymes work in the acidic pH. The digestion of protein starts in the stomach. The food stays approximately 3 to 4 hours in the stomach, and then it goes into the small intestine. When it goes into the small intestine, it gets two secretions. One, from the gallbladder, it receives the bile. Bile is made in the liver and stored in the gallbladder. The bile, it emulsifies the fat and neutralizes the stomach acid pH, which comes up with the food. And then the pancreas releases the pancreatic juice, which contains all the enzymes to finish up the digestion. So all carbohydrates, proteases, and lipase are present in this pancreatic juice. The digestion is finished. In the small intestine, the food gets absorbed into the blood, and then undigested food passes into the large intestine. The large intestine absorbs all the water and converts the undigested food into the semi-solid mass called the feces, and the feces is thrown out of the body through rectum or the anus. So this is the whole process of digestion. If you see this, we can break this digestion process into 5 stages. One is ingestion. Ingestion is the taking in food. It helps with the help of the mouth and the tongue. The food is churned, so mechanical digestion takes place, and the food is mixed with the saliva. Then is the digestion. It starts in the mouth, and stomach, intestine, pancreas, and liver are involved in it, which is the breakdown of large insoluble particles into smaller ones. Then in the small intestine, absorption, the process of digested food are absorbed into the blood. Then assimilation happens in the body cell, where this absorbed food is used for the body processes. And there's an ingestion, egestion, which is the getting rid of undigested food material, which happen through rectum and anus. Okay, now let us just quickly summarize this entire digestion process. So it starts with the mouth, where food is ingested, mechanical digestion starts, and amylase is mixed, which starts to break down the starch into the sugars. Then it is the pharynx, which is at the beginning of the throat, and both food and air travels to it. Then there's a small flap called epiglottis. Epiglottis is a flap of muscles that closes the windpipe so that the food does not get into the windpipe, and it directs the food into the esophagus. Esophagus is the tube that leads the food into the stomach, and it has some peristalsis movement that pushes the food down. Then the food comes into the stomach, where the mechanical digestion and the chemical digestion starts. The hydrochloric acid is released, and an enzyme called pepsin is released that digests proteins into amino acid. Then the food goes into the small intestine. There, the liver that makes the bile stores into the gallbladder. The secretion of the bile comes into the food that neutralizes the food, and then gallbladder also releases bile, which is stored in the gallbladder. And then the small intestine, where the digestion finish and the absorption starts. And then there's a small organ here called the pancreas, which releases the pancreatic juice. And yeah, pancreas, it is where it produces the pancreatic juice that contains the lipases, proteases, carbohydrates enzyme. Then the food goes into the large intestine, which absorb all the water. Then it is thrown out of the body through the rectum and anus. So you should know all the digestive organ. You should be able to label them in the exam, and you should know the function of each of the parts. Okay. Now then, the very important function is the bile juice. It is a greenish yellow alkaline liquid which is produced in the liver and stored in the gallbladder, and it performs two major functions. You should remember that. One is the neutralization. So the food that comes from the stomach is acidic, and the enzyme of the intestine can only work in alkaline condition. So the bile neutralizes the food that comes from the stomach and makes it alkaline, so that enzyme release in the intestine can work effectively. Next is the emulsification of the fats. For lipase to work, the fat must be broken down into smaller droplets to increase the surface area for the lipase to function. The bile performs this function of emulsification of fat for the efficient working of the lipase. Now, let's see what are the components that we take in with the food. We take carbohydrates, proteins, and fats. Carbohydrates are present in bread, pasta, cereals, and rice. Proteins are present in pulses, chicken, meat, poultry, egg, beans, and nuts. And fat is present in butter, oil, margarine. Carbohydrates are composed of sugars. Proteins are made up of amino acids, and fats are made up of fatty acids and cholesterol. And this is what these components get digested into. The enzyme for carbohydrates are carbohydrates. Example, amylase. For protein, it is proteases, and for fat, it is lipases. Carbohydrates are digested in mouth and small intestine. Proteins are digested in stomach and small intestine, and fats are digested just in small intestine. Carbohydrates are the principal source of energy. They are fuel for respiration. They can be storage molecules like starch and glycogen, or structural molecules like cellulose in plants. Proteins are the components of muscles required for growth and repair. They're the components of enzymes and they are also components of some of the hormones like insulin. Fats are used to insulate the body. They are the reserve source of energy, and they also makes the cell membrane. Okay, so you should know what are the different components of food, their sources, where they're digested, and what is their function in the body. Okay, so now since we are doing digestion, and digestion is chemical, that is, it involves the use of enzyme, you should know what are enzymes. So make sure you remember this definition and this lock and key model, very, very important. So I'll just take you through it. So enzymes are biological catalysts. So I think in chemistry, you remember what are catalysts? Catalysts are those substances that increases the rate of reaction without being used up. So if there's a catalyst which are used in biological reaction, we call them enzymes. So enzymes are biological catalysts that increases the rate of a biological reaction without being used up. Now, what do enzymes do that increases the rate of reaction? They actually provide an alternative route to a reaction, which works by lowering the activation energy. And enzymes are chemically proteins, so they are very sensitive to heat, pH, and temperature. Okay, now this is the structure of the enzyme, and the enzyme has a specific shape, and it has a site called the active site. Active site is a site where substrate molecules bind to it. Now, substrate and the active sites, they have complementary shape. Remember to use the word complementary. Most of the time in the exam, students say substrate and active site have same shape. No, that's wrong. It's like a lock and key. The substrate and the active site, they have a complementary shape. The substrate binds to the active site, forms an enzyme substrate complex. The enzyme substrate complex react to form an enzyme and the product, and it disintegrate to give the product back, and your enzyme gets regenerated. So remember, enzymes are highly specific due to the active shape. As the active shape has a specific shape which is complementary to the substrate, so the specific substrate molecule can fit into the active site of the enzyme. Now, what are the factors that affects the enzyme activity? Temperature and pH are one. We'll be discussing that. But the substrate concentration and enzyme concentration also affects the rate of reaction. So, as we increase the substrate concentration, there'll be more molecules to bind with the active site. Therefore, the rate will increase at the start. But after that, it levels off because all the enzyme active sites will be used up, and then enzymes will become a limiting factor. The same thing works with the enzyme concentration. As we increase the enzyme concentration, there will be more active sites for the substrate molecules to bind to. But after that, further increasing the enzyme concentration will have no effect because then all the substrate molecules will be used up. So substrate will become limiting. So the graph will level off. Now for the temperature, as we increase the temperature, the rate of enzyme catalyzed reaction increases because the particles gain kinetic energy. As per chemistry, they'll collide more the collision, more the chances of reaction. It increases up to a point, and this point is called the optimum. Optimum is the point where the enzyme activity is the highest. But after that, the rate will drop suddenly. Why is that? This is because the enzyme gets denatured. Now, what is denaturation? Now, denaturation is when you heat any specific enzyme due to being protein in nature, their bonds break. So they change the shape. Now, when they change the shape of the active site. So the substrate molecules can no longer bind to it, decreasing the rate of reaction. So denaturation is change in shape of the enzyme with a change in pH or the temperature. The same effect happens with the pH. As you increase the pH, it increases the rate of reaction up to the optimum, and after that it drops. It doesn't drop that suddenly like temperature, so we make like a bell-shaped curve. And then it again drops. Why? There's a denaturation because the shape of the enzyme changes. Now for the temperature, the optimum is around 37 to 40 degrees because that is the normal body temperature. But for the pH, different enzyme has different optimums. Like stomach enzymes, they work best in acidic condition, and the intestinal enzymes, they work in the alkaline condition. Okay, so I hope this graph is clear to you. You should be able to draw this graph in the reaction in the exam, or if this graph is given, you should be able to explain all this trend, why there's an increase, what is an optimum, and why there's a drop. And I hope you will remember it. Now let's move on to a circulatory system. The question is, why we need a circulatory system? And what is a circulatory system? So circulatory system is a system that provides the body with oxygen, nutrients, hormones, or all the essential things that a body needs to survive. And it also gets rid of the waste products. But why do we need a circulatory system? We need a circulatory system because we need constant supply of all these nutrients. And circulatory system consists of three components: a pumping organ, an organ that pumps the transporting system, transporting medium. Transporting medium is the blood, which has all the things that a cells required. It is pumped by a pumping organ called the heart, and the blood is taken from one cell to another, or from one organ to another, to the system of vessels, which is arteries, veins, and capillaries. So now we'll be looking over what is blood, heart, and these arteries, veins, and capillaries. Now, if you talk about the blood is made up of blood plasma and blood cells. So if you see blood, we centrifuge the blood, then the blood cells are heavy, they collect at the bottom, and then there's the blood plasma. Blood plasma makes up 55% of the blood, and it is the liquid component of the blood, and it has all the things that a cell requires, like glucose, amino acids, hormones, waste product, carbon dioxide. And then the blood cells, they perform specific function, and there are three types of blood cells: red blood cells, white blood cells, and platelets. The function of the red blood cell is to transport oxygen all around the body, and it's a special cell. Why it's a special cell? Because to carry out these special functions, it has special adaptations. Like, they are in the form of biconcave disc, which increases the surface area for oxygen transport. They have no nucleus, so that they can have more room for the oxygen, and they contains a pigment called the hemoglobin, which binds with oxygen and carry it all around the body. Then you have the white blood cells. White blood cells are the soldiers of the cell. They protect the body from infections. They are made up of two types of cell: lymphocytes, they produce antibodies, and phagocyte, which engulfs the pathogen. And then we have the platelets. They are involved in blood clotting. They have fibrin proteins, which form mesh around the blood and prevents the excess blood from flowing out by forming a clot. So I hope you will remember what are the different cells and their function, and what is the function of the blood plasma, and what are the different components of the blood. Okay, so now we will talk about the heart, but before that we should know why we need a circulatory system. We need a circulatory system because we have low surface area to volume ratio, we are metabolically active, and we are multicellular. Now, low surface area to volume ratio because we have a bigger size. As the size increases, the surface area to volume ratio decreases. And at such a low surface area to volume ratio, diffusion alone is not effective to meet the growing need for the body. Therefore, we need a circulatory system. Now, being metabolically active, we need a constant supply of oxygen, glucose, and nutrients. And at the same time, the waste product is also made at a greater level. So we need a constant removal of waste products. So we need a circulatory system to do that task. And being multicellular, we have lots of cells which are deep inside. So diffusion distance is too large. So we need a circulatory system to transport substances deep inside the cells as well. So therefore, we have this circulatory system, and the circulatory system we have is double circulation. Now, let me explain you how this double circulation works. Now, in our circulation, we have a lungs and the heart is 4 chambers. The top chambers are the atriums, and the bottom is the ventricle, and our right is the heart left. So this is the left atrium, right ventricle, right ventricle, and the body. Now, how does the circulation start? The lungs gives oxygen to the blood, and the oxygenated blood from the lungs, it starts flowing to the left atrium. The left atrium then pushes the blood to the left ventricle, and then left ventricle pushes the blood out, and it goes to all parts of the body. The body takes oxygen and all the required nutrients from the blood, and then it releases carbon dioxide into the blood, and the blood becomes deoxygenated. This deoxygenated blood from the body is pumped back into the right atrium. The right atrium takes the blood to the right ventricle, and the right ventricle then takes it back to the lungs so that it can pick some more oxygen, and the cycle continues. Okay, now if you see the circulation, it is consist of 2 circuits. One, lung to the heart and heart to the lung, and that is known as pulmonary circulation, and heart to the body and body to the heart, that is known as systemic circulation. And both of these circulation together make a double circulation. So that is one of the definition of double circulation, that it consists of 2 circuits, a pulmonary and the systemic. The second way to describe this double circulation is, this is circulation in which the blood is pumped to the heart twice in one complete cycle. So in one complete cycle, the blood is pumped to the heart twice. It involves 2 circulation, pulmonary, which is between lungs and the heart, and systemic, which is the circulation between heart and the body. The double circulation makes the circulation more efficient by preventing the mixing of oxygenated blood, and helps to alter the pressure from the different chambers of the heart. So here you can see in double circulation, the oxygenated blood is all to the left side and deoxygenated blood to the left side, and the heart can modulate the pressure. When it is sending the blood to the body, it can increase the pressure, and when it is sending the blood to the lungs, it can decrease the pressure, and that can only be done as it has a double circulation. Now let us label the vessels. Now, if to label the vessels, always remember anything that gets the blood to the heart will be the vein, and anything that takes the blood away from the heart is an artery. So in the first case, the blood is coming to the heart from the lungs. So this has to be a vein. And since it is the vein associated with the lung, so it has the word pulmonary vein associated with it. So the blood comes from a pulmonary vein to the left atrium. Then it goes to the left ventricle, and then from the left ventricle, it goes to the body. So blood is going away from the heart. So this has to be an artery, and it has to be a largest artery, as it is giving the most difficult task of sending the blood to all parts of the body. So this is aorta, which is our largest artery. Then the blood comes from body to the heart. Again, it has to be a vein. So it's a vena cava. And then from the right ventricle, it goes back to the lung. So it has to be the artery. And since it is associated with the lung, so it is the pulmonary artery. So you should be able to draw this circuit and label all the vessels. And I hope this is clear to you. Next is we have this heart. Heart is a pumping organ, works all day and night. It has 4 chambers. Atriums are the top. So this is left atrium. This is right atrium. The ventricles are at the bottom. So this is left ventricle, the right ventricle. Ventricles are doing the biggest job as they are pumping the blood to a greater distance. So you can notice that atrium has a thin wall and ventricle has the thickest wall. And as compared to left ventricle and right ventricle, left ventricle has to send the blood to all parts of the body. So it has to send the blood at a very high pressure. So it has the thickest wall. Okay, and then the left atrium receives the blood from the pulmonary vein. It goes to the left ventricle. The left ventricle sends the blood to all parts of the body. So that's the aorta. The blood travels all parts of the body and come back to the right atrium through the vena cava, and from the right ventricle, the blood pump back to the lungs via pulmonary artery. So you should know the structure of the heart and the labeling and the functions of all the parts, and I hope this is clear to you. Now, if you remember that in a circulatory system, we need the blood vessels that carries the blood all around, and there are 3 types: arteries, the capillaries, and the veins. The capillaries connects arteries and veins, and what you can see in arteries and vein, the lumen of the vein is bigger and the lumen of the artery is smaller. On the other hand, the artery is surrounded by a thick muscular tissue, which is not present in the capillaries, and veins have thin. So let's tabulate the difference between them and make sure you remember all these differences and able to write in the exam. Arteries carries the blood away from the heart. Veins carries the blood to the heart. Capillaries connect arteries and veins. Arteries, blood flow at a high pressure. In veins, it flow at a lower pressure, and capillaries flows at a lower pressure. So to withstand high pressure, the arteries have thick muscular elastic wall. Veins have thin walls, and capillaries are 1 cell thick. Arteries have a narrow lumen, veins have a wider lumen, and capillaries have a very small lumen. As the blood flows at a very low pressure in veins, there are chances that the blood can backflow. So veins have valves to prevent the backflow of the blood. Arteries and capillaries do not have valves. Arteries generally carry the oxygenated blood except the pulmonary arteries. Vein carries deoxygenated blood except the pulmonary vein. And the capillaries carries both oxygenated and deoxygenated. So I hope you are able to spot by the diagram whether it's in arteries, veins, and capillaries with their function. Okay, now since we have discussed the heart, there are a lot of problems that can come up with the heart, and they are known as coronary heart diseases. Now, what are coronary heart diseases? If there's a problem in a coronary artery. What's a coronary artery? It is an artery it's the branch of the aorta that supplies the blood to the heart. And if these arteries become narrow, why they become narrow? You can see this is a normal coronary artery, and in the coronary artery there is a plaque that is deposited. What is a plaque? It is a fatty materials like cholesterol which get deposited. It narrows the artery. The narrowed artery reduces the blood flow to the heart. The blood flow to the heart is reduced. The heart do not get enough oxygen. It can cause heart pain, chest pain, and heart attack. So this is what is the plaque deposition that happens in a coronary artery. Now, how we can improve that? We can insert a stent. A stent is placed which has a balloon with it into the blocked artery. The balloon is inflated and the balloon opens the blocked artery, and a mesh stent made up of metal is placed which holds the narrow artery and prevent it again from collapsing, and people are also given the medicine called the statins, which lowers the blood cholesterol and prevents the further deposition. So this is one of the way, and this is also known as angioplasty, in which a balloon or the stent is used to open the blocked artery. But there are cases where the stent cannot be performed due to maybe the blockage is too much or some of the problems to be done a patient can have with the stent. Then in that case we do a bypass. What's a bypass? See, you can see that this is a blocked artery. We give a graft, and this graft bypass the blocked artery. Okay, so there's a blockage in the coronary artery, and artery graft is stitched to the coronary artery, so the blood, rather than flowing through the blocked artery, it flows to the graft and it resumes the blood flow. Okay, now there are another problems as well apart from this that can be associated with the heart. These problems are like a leaky valves. A person can have leaky valves, maybe by time that the blood flows at a very high pressure, so the person can develop leaky valves. So due to increased pressure of blood flow, the valve starts to leak. For that we have valve replacement, where the valves made up of metals or polymers are placed, or the biological valves from the animals can be used. At times the person get irregular heartbeat. Why? Because the heart is controlled by the groups of cells present in the left atrium called the pacemaker, which regulates the heartbeat. If there's any problem with that, there's an irregular heartbeat, so we make electrical devices called artificial pacemaker, which takes over the function of the natural pacemaker, and they regulate the heartbeat. Then apart from all this problem, the person can have a greater damage to the heart or heart failure. In that case, we need a heart transplantation or artificial heart, which is not that successful, but the research is going on, but heart transplant can be done. So these are all the non-communicable diseases, or you can say the problems with the heart, and the proposed treatment for the same. Okay, now after the circulatory system, we come to the human gas exchange system called the respiratory system, which starts with the nose, and then the air goes into the windpipe. The windpipe is known as the trachea. The trachea is covered by the ring of cartilages that prevents the trachea from collapsing, and then the trachea bifurcates into two. These two bifurcations are known as the bronchus. These two bronchus enters the lungs. In the lungs, the bronchus gets further branched into the bronchioles, and at the tip of the bronchioles, we have greater air sacs called the alveoli, where the gas exchange takes place. And then there's a dome-shaped muscular organ called the diaphragm, which goes up and down to regulate the chest volume for inspiration and expiration. Now, what is the function of the respiratory system? Breathing system is to breathe. Breathe is taking in oxygen and giving out carbon dioxide. Now, for the air to come in, air always move from high pressure to a low pressure. So since the air coming in, the pressure inside the lung is low. For expiration, the pressure inside the lung is high. If the pressure is low, the volume of the lung should be high, and the volume of the lung should be low in expiration. How these volume increases in inspiration? It is the muscles, the ribs muscle, they contract and the rib moves outwards. In expiration, the ribs relaxes and the rib move downwards. The diaphragm, it flattens, which increases the volume and the air moves in, and expiration, the diaphragm domes up, which decreases the volume and the air pushed out. So you should know what is the mechanism of inspiration and expiration, and what changes takes place inside the lungs or the ribs for inspiration and expiration to take place. For gas exchange, if you remember, we talked in cell biology, there's an alveoli which has a greater surface area by having a special shape and being millions of them. They have a shorter diffusion distance by being one cell thick, and they maintain a steep concentration gradient by having a rich blood supply. By all this factor, alveoli has adapted themselves for an efficient gas exchange. Now we have discussed organization in animals. Now let's discuss organization in plants. The plant cell is there. We discussed the plant cell in the cell biology video. Then the plant cell get organized into tissue, xylem, phloem, these palisades and spongy mesophils. These are the tissues. You can see sponges are the ones which have greater air spaces between them but are photosynthetic. The palisade cells are arranged next to each other, which are also photosynthetic. In the organ system, we have a water transport system and the food transport system. Water transport system consists of xylem and the roots. The food transport system consists of the phloem and the leaves, and all of this make a plant organism. Okay, now what is a transpiration? Transpiration is the loss of water from the surface of the leaf in the form of water vapor, and this happens to a stomata. So stomata are these air spaces or air pores present on the surface of the leaf, which consist of stomatal pore and these cells, which are known as the guard cell. The guard cells open and closes, which controls the water movement, and the gas exchange also takes place to the stomata. Now, this transpiration makes the water uptake or the water transport system. How? Water is lost from leaf pore in the form of transpiration. This decreases the concentration of the water, so the water from the neighboring cell moves in. Now, as the water from the neighboring cell moves in, that decreases the concentration of water in that area. So it takes water from the neighboring cell and then from the neighboring cell, which makes a long continuous column, and eventually it forces the water from the soil to comes into the root, and then the xylem carries up due to transpiration stream. Okay, now what are the factors that affects transpiration? It is temperature, light, wind, and humidity. With increase in temperature, rate of transpiration increases because the particles gain kinetic energy. They start to evaporate faster. With light, rate of transpiration also increases because remember, as the photosynthesis will increase, the transpiration will increase, because as the rate of photosynthesis increases, stomata will increase to take in more carbon dioxide, which increases transpiration. Wind, as the wind speed increases, it moves all the saturated air, increasing the concentration gradient of the water vapor, increasing the rate of transpiration. Humidity, as the rate of transpiration decreases with humidity. Why? Because humidity will make the air saturated with water vapor, decreasing the concentration gradient, so the diffusion rate of water will slow down. Okay, now how do we measure the rate of photosynthesis or transpiration? I think you must have done this experiment. This is a potometer. A potometer, there's a glass tube, there's a reservoir of water, and there's a beaker, and there's an air bubble. As the water uptake, as the plant will lose water, it will try to uptake this water, and this bubble will move. You will use a scale and measure how much the bubble is moved. The movement of bubble is an indicator of water movement, which in turn shows the rate of transpiration. So we assume all the water that it is uptaking is equivalent to the rate of transpiration of water. Okay, now since transpiration is a necessary evil, why? Because transpiration is required for the uptake of water, but it can also cause the plant to unnecessarily lose the water and can wilt, they can droop. So the plant has done an adaptations to limit water loss. They are covered with the cuticle layer, which is waterproof and prevents the rate of transpiration. They have stomata on the lower surface that reduces the rate of transpiration. Some plants' stomata opens at night and close during the day. In some plants like cacti, the leaves are reduced to spines that limits the rate of water loss. And some plants have sunken stomata, where the stomata is in pits, and they have the humid air around it that prevents the rate of transpiration. Okay, so I hope this unit is clear to you. Now it's the time to test yourself. In this unit, we have seen a lot of key terms. So you should be able to explain these key terms. So these key terms are cell, tissues, organs, organ system, digestive system, esophagus, stomach, liver, pancreas, small intestine, large intestine, substrate, active site, carbohydrates, protein, fats, enzyme, catalyst, denaturation, amylase, protease, lipase, bile, emulsification, plasma of the blood, red blood cell, white blood cells, platelets, plasma, arteries, veins, and capillaries, double circulatory system, coronary artery, coronary heart diseases, stents, statin, pacemaker, xylem, phloem, transpiration, translocation, potometer, stomata, and gut cell. So you can pause the video, have a go at these terms, and then check yourself if you know these terms. You can come back to my website. The link is mentioned in the description box below, and you can have the answers of them as well. Okay, so what is the next step? Next step is check the specification. Make sure whatever thing is there in the specification is crystal clear to you, and do exam questions on this topic. Okay, so I hope you have understood this video well. If you still have any doubts, leave a comment below. We'll reply you as soon as possible. Or you can go to our website where we have 24/7 chat support, where you can post any queries before your exam and you can get instant reply. You can go to my website where you can find the notes of this as well. The link is mentioned in the description box below. If you like this video, then choose subscribe to our channel and do not forget to like, comment and share this video. And if there's any specific topic you want me to make a video on, do leave a comment below and I'll make sure I'll put that up before your exam. Okay, so please go over this topic very, very seriously. It's a very important topic and I'll see you next in your next video. Till then, happy revising.